Hello, my name is Mahesh Vallabhaneni and this is my video entry for the 2019 Big Bang competition. As humans, we perceive up to 80% of our impressions through eyesight, arguably making sight the most important of all senses. Loss of vision can be devastating to anyone. A condition called glaucoma is the leading cause of irreversible blindness worldwide. Currently, there are estimated to be 64.3 million people who are suffering from the condition across the globe. Unfortunately, more and more people are affected each passing year, and as shown by the graph enclosed in File 1, a recent study has estimated that by the year 2040, 111.8 million people will suffer from the condition. Furthermore, the study conveyed that there would be a greater impact on the African and Asian regions that already suffer due to a lack of structured public health. Therefore, further research into prevention and developing new treatments is crucial to allow enough time for these regions to adopt a public health strategy to combat the problem effectively. I was delighted to have had the opportunity to join a research team at the University of Liverpool to assist in the project centred around investigating a link between glaucoma and the overexpression of a gene known as Connective Tissue Growth Factor, or CTGF for short. I came across this opportunity during work experience in a hospital, where I became fascinated by imaging within medicine, such as MRI scans and the complex use of X-rays in the field of interventional radiology. Since then, I've been keen to learn more about imaging from a molecular or cellular level all the way up to a clinical level. Having interacted with people suffering from glaucoma during volunteering, glaucoma itself was a condition I wanted to gain a greater understanding of. The project provided me with the perfect opportunity to further my knowledge and understanding of cellular level imaging through performing techniques such as a hematoxylin and eosin stain, also known as H&E staining. Understanding and researching this specific staining process constituted the bulk of my research experience. CTGF is a matricellular protein, which means that it is a non-structural protein which is found in the extracellular matrix. The protein is associated with cell adhesion and fibrosis. Fibrosis is the thickening and scarring of connective tissue. This fibrotic quality of CTGF was theorized to cause an increase in the intraocular pressure of the eye and subsequently result in damage to the optic nerve, causing irreversible blindness. By examining and comparing slides under a microscope between mice which had an overexpression of CTGF induced within them compared to mice which had no such overexpression, we hoped to see whether there was a clear difference in the damage to the optic nerve. In order to clearly observe the damage, we used H&E staining. As mentioned earlier, the H&E staining process and microscope imaging was the main focus of the project. I researched and revised the intricate steps required for the process. There was a need to be precise and time effective in the staining in order to fit into the period of time that was allocated within the lab. This required preparation to make sure all of the equipment and necessary chemical reagents were prepared. The steps involved firstly removing the paraffin wax surrounding the slides by washing the slides in xylene for 5 minutes. The slides were then washed again in another pot of xylene for an additional 5 minutes. The slides then underwent a total of 15 minutes of ethanol washing. This consisted of 3 sets of 5-minute washings in various ethanol solutions. We used 100%, 90% and 70% ethanol solutions in that respective order. The slides were then rinsed in water and ready for hematoxin application. We applied hematoxin for a total of 4 minutes. Immediately after this, the slides were rinsed in water and dipped in a 1% acid alcohol solution. The slides then spent another 5 minutes in water. This was followed by them being washed in alcoholic eosin for 5 minutes and a short rinse under running water. The dehydration process of slides required a 90-second ethanol washing, which was split into 30-second periods in 3 different concentrations, the first being 70%, then 90%, and finally 100% ethanol. The slides were then washed twice in xylene for a minute each time. Finally, the cover slips were mounted using a DPX mountant, making sure not to get any air bubbles over the tissue. These slides were then left overnight to dry for imaging in the morning. The slides were delicate, and therefore precision was key to ensure the specimens did not fragment. We prepared a large number of slides in case a few did fragment. The next day, I imaged the slides using a powerful microscope. This process gave me an appreciation of how to operate an advanced microscope, which was an interesting skill to learn. The colouring of the H&D staining was magnificent to see, and I managed to focus in on the optic nerve and surrounding areas. This concluded the majority of the laboratory work for the project. The remainder of the research prior to analysing the results was oriented around researching more into the H&D staining and glaucoma itself. I was curious to find out how the H&D staining worked. Research led me to uncover that the hematoxin is largely classified as a basic dye when used in conjunction with the mordant. The mordant is a substance which is usually an inorganic oxide that combines with the dye to fix it to a material. Hematoxin is comprised of a dye known as hematin, which is obtained from the logwood tree, and the addition of aluminium ions. The hematoxin had an affinity for acidic structures which are considered basophilic, such as DNA. A linkage is created between these basophilic structures and the hematoxin through a mutual binding to the mordant, which in this case was the aluminium. This leaves these structures in a range of shades from purple to blue. Eosin acts as a counterstain. It is an acidic dye and therefore has an affinity for basic or acidophilic structures such as cytoplasmic filaments in muscle cells. This stains these structures in a range of shades from red to pink. The next part of the research was to understand more about glaucoma. There are two main types of glaucoma: primary open angle glaucoma and primary acute closure glaucoma. The former is a condition which worsens over a long period of time, whereas the latter is a rapid onset condition leading to a fast increase in intraocular pressure. The first of the two, the primary open angle glaucoma, is the type which this project focused on. Glaucoma is becoming an increasing problem within this country, as it is more prominent among those over 70, and therefore the NHS is experiencing more glaucoma cases with an ageing population. I was fascinated by medical literature which outlined the predicted progression of glaucoma by 2040. I referenced to these figures earlier. The treatments for glaucoma are usually eye drops to relieve intraocular pressure, or laser eye treatment to open up blocked drainage canals in the eye. Surgery can also be used to achieve the same outcome. The link between glaucoma treatment and the project is something I will discuss further after explaining the outcomes of the project. Files 2 and 3 are just two examples of the images I took of the slides using a Nikon CI microscope. The yellow circled regions show a severely deteriorated optic nerve with CTGF overexpression, compared to the normal optic nerve with no CTGF overexpression. The optic nerve appeared fragmented in nature and lacked fullness. This can be best observed through the sloping nature of the optic disc, which I've highlighted using a green oval. This supports the idea that the overexpression of CTGF does in fact contribute to the primary open angle glaucoma. This was the link which we had hoped to identify by carrying out the investigation. As I mentioned, the threat of glaucoma is becoming a more serious one, and it is imperative to find multiple and effective treatments due to the added time pressure caused by the likelihood that more time will be needed to create effective public health strategies in the less developed areas of the world, which are the most likely to be the most heavily affected. The link between CTGF and glaucoma invites the possibility of more research into the linkage between not only CTGF but also other genes and glaucoma. This creates the prospect that in the future, effective gene therapies could potentially be created as an option for treating glaucoma, as opposed to invasive surgery, which often carries risks and complications. These risks and complications are heightened for the elderly demographic which suffer the most from primary open angle glaucoma. More research needs to be carried out to identify the exact reasons why CTGF contributes to primary open angle glaucoma. I wish to thank those at the University of Liverpool for supervising me and helping me out a lot with this project, and I also wish to thank you for taking the time to watch this video.